Doctor, can you explain Mendel's contributions to genetics and his key experiments?
Certainly. Johann Gregor Mendel, often called the father of genetics, made groundbreaking discoveries in heredity during the mid-19th century. Despite being more recognized as a meteorologist in his time, Mendel's experiments with pea plants laid the foundation for understanding how traits are passed from one generation to the next. Mendel studied characteristics like flower color, seed shape, and plant height, focusing on traits influenced by single genes. He published his findings in the book Experiments on Plant Hybrids in 1865. Interestingly, the terms gene and allele were not known during his era. These concepts were developed later.
How did Mendel conduct his experiments?
Mendel chose pea plants because they typically self-pollinate, resulting in highly homozygous offspring. This allowed him to observe the inheritance of specific traits without external interference. He crossed plants with contrasting traits, such as tall versus short stature or purple versus white flowers, and meticulously recorded the outcomes.
What were the key principles Mendel discovered?
Mendel formulated 3 fundamental laws of inheritance. First law, the law of uniformity of F1 hybrids. When 2 homozygous parents with different traits are crossed, all the offspring in the first generation, F1, are genetically uniform and display the dominant trait. For example, crossing a plant with purple flowers, AA, with one with white flowers, AA, produces F1 offspring with purple flowers, AA. Second law, the law of segregation. During gamete formation, the 2 alleles for each gene segregate so that each gamete receives only one allele. When heterozygotes, AA, form gametes, half carry the dominant allele, A, and half the recessive, A. The Punnett square illustrates this, predicting a 1:2:1 ratio of genotypes, AA, 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 and a 3:1 phenotypic ratio in the F2 generation. Third law, the law of independent assortment. Alleles of different genes assort independently into gametes when the genes are located on different chromosomes. This law explains the 9:3:3:1 phenotypic ratio observed when tracking 2 traits simultaneously, such as seed color and shape.
Are there exceptions to Mendel's laws?
Yes, exceptions occur in several scenarios. Polygenic inheritance, where traits are influenced by multiple genes. Linked genes, located close together on the same chromosome, often inherited together. Gene interactions, where one gene modifies the effect of another. Non-Mendelian inheritance, like mitochondrial DNA, which is passed exclusively from the mother.
Can you explain dominance relationships?
Certainly. Mendel observed complete dominance, where the dominant allele entirely masks the recessive one. However, there are variations. Incomplete dominance. Heterozygotes show a blend of traits, like pink flowers from red and white parents. Codominance. Both alleles are fully expressed, as seen in AB blood type. Recessivity. The recessive trait appears only in homozygous individuals, like white flowers in AA plants.
What about testing hypotheses in genetics?
Mendel employed methods like the chi-square, chi-superscript 2, test to compare observed and expected outcomes, verifying his predictions. This statistical approach ensures that results align with theoretical models.
Could you summarize Mendel's significance?
Mendel's work revolutionized biology by providing a framework for genetic inheritance. Though his contributions were unrecognized during his lifetime, scientists like Hugo de Vries and Carl Correns rediscovered his work in the early 20th century, validating his insights. Today, his principles remain fundamental in genetics and continue to inform research across biology and medicine.
That was fascinating. Did Mendel also explore any complex inheritance patterns beyond the basics?
Mendel's original work focused on straightforward, single-gene traits. However, modern genetics has expanded on his principles to understand more complex inheritance patterns, such as epistasis, where one gene can suppress or modify the effect of another. Polygenic traits, like height and skin color in humans, influenced by multiple genes. Gene linkage, discovered later by scientists like Thomas Hunt Morgan, where genes located close together on a chromosome do not assort independently due to reduced recombination between them.
You mentioned dihybridism earlier. Could you elaborate on that?
Of course, dihybridism examines the inheritance of two traits simultaneously. For example, Mendel studied seed color, yellow or green, and seed shape, smooth or wrinkled. Crosses between heterozygotes for both traits, ABB, produce a phenotypic ratio of 9:3 to 3:1 in the F2 generation, assuming the genes are unlinked. Mendel's third law, the law of independent assortment, applies here, stating that the inheritance of one gene does not affect the inheritance of another if the genes are on different chromosomes. However, this breaks down when genes are linked.
Can you clarify the role of crossing over in genetics?
Certainly. Crossing over occurs during meiosis, where homologous chromosomes exchange genetic material, creating new combinations of alleles. This process increases genetic diversity in gametes. When two genes are physically close on the same chromosome, crossing over between them is less likely, leading to genetic linkage. The frequency of recombination is used to map genes on chromosomes, measured in centimorgans, cm.
What about traits influenced by gender? Are those inherited differently?
Yes, sex-linked traits follow unique inheritance patterns. For instance, X-linked recessive traits, e.g., hemophilia or color blindness, are more common in males because they have only one X chromosome. Y-linked traits are passed exclusively from father to son, like the SRY gene, critical for male development. In addition, some traits, while autosomal, are sex-limited or sex-influenced, meaning their expression depends on an individual's sex. For example, baldness is a sex-influenced trait, and milk production in mammals is sex-limited to females.
It's incredible how these principles apply broadly. Are there real-life examples of Mendelian traits?
Absolutely. Many human traits follow Mendelian inheritance, such as dominant traits, the ability to roll one's tongue or having widow's peak hairlines. Recessive traits, albinism or attached earlobes. Beyond humans, Mendel's laws also explain patterns in plant and animal breeding, helping us select for desired traits like high-yield crops or livestock.
Mendel's contributions seem so fundamental. Are there any modern tools that directly stem from his work?
Definitely. Tools like the Punnett square for predicting offspring ratios and statistical tests like the chi-square analysis are direct applications of Mendel's principles. Furthermore, his insights underpin genetic counseling, breeding programs, and even advanced technologies like CRISPR gene editing. His foundational work reminds us of the importance of observation and experimentation in science. Though Mendel's findings were overlooked for decades, they eventually became the cornerstone of modern genetics, influencing fields from agriculture to medicine.
Doctor, earlier, you mentioned genetic linkage. Can we dive deeper into how this concept works?
Of course. Genetic linkage occurs when two genes are located close together on the same chromosome, and they tend to be inherited together rather than assorting independently. The closer the genes, the stronger the linkage because crossing over is less likely to separate them during meiosis. To quantify linkage, we use the recombination frequency, expressed in centimorgans, cm. 1 centimorgan represents a 1% chance of recombination between two genes. If the recombination frequency exceeds 50%, the genes are likely unlinked and assort independently.
How is genetic linkage tested in experiments?
Geneticists perform test crosses using dihybrids and recessive homozygotes. By analyzing offspring ratios in the next generation, we can determine the recombination frequencies and construct linkage maps of chromosomes. This method was pioneered by Thomas Hunt Morgan in his studies with fruit flies.
Are there examples of linkage influencing human genetics?
Yes, a classic example is the study of hemophilia and color blindness, both X-linked traits. When these two traits are inherited together more often than expected, it suggests they are linked on the X chromosome. These insights are vital for understanding hereditary diseases and for mapping the human genome.
You mentioned earlier that Mendel's laws don't always apply. Can we discuss when they don't?
Certainly. Mendel's laws apply mainly to traits influenced by single genes with clear dominant and recessive alleles. However, deviations occur in polygenic inheritance, traits like height and skin color, which result from the interaction of multiple genes. Gene interactions, such as epistasis, where one gene affects the expression of another. Non-Mendelian inheritance includes mitochondrial DNA passed maternally and genomic imprinting, where only one parent's allele is active due to epigenetic modifications. Environmental factors also play a role in modifying phenotypes, adding another layer of complexity to inheritance.
What about traits influenced by chromosomal abnormalities?
That's a great question. Chromosomal abnormalities, such as deletions, duplications, or translocations, can disrupt Mendelian patterns. For instance, Down syndrome results from an extra chromosome 21, trisomy 21. Turner syndrome occurs when a female has only one X chromosome. Klinefelter syndrome arises in males with an extra X chromosome, XXY. Such conditions often lead to complex phenotypes not predictable by simple Mendelian laws.
Finally, could you explain how Mendel's work influences genetics today?
Mendel's principles form the foundation of modern genetics, influencing everything from agriculture to medicine. His methods are still used to predict hereditary traits, diagnose genetic disorders, and develop breeding programs. In research, his laws guide studies on gene function and inheritance patterns. Even advanced technologies, like genetic engineering and genome editing, owe their conceptual roots to his discoveries. By laying the groundwork for understanding inheritance, Mendel transformed biology into a predictive science. His legacy reminds us of the power of curiosity and careful experimentation.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.